Welcome to Lesson 15.3, Applications of Genetic Engineering. In this lesson, we'll describe the benefits of genetic engineering as they relate to agriculture and industry, explain how recombinant DNA technology can improve human health, and summarize the process of DNA fingerprinting and explain its uses. Do you think you've ever eaten genetically modified foods? If you've eaten corn, potatoes, or soy products, chances are close to 100% that you've eaten food modified in some way by genetic engineering. Ideally, genetic modification could lead to better, less expensive, and more nutritious food, as well as less harmful manufacturing processes. Let's discuss the Bt toxin. While this toxin is harmless to humans and most other animals, enzymes in the digestive system of insects convert Bt to a form that kills the insects. Plants with the Bt gene, then, do not have to be sprayed with pesticides. In addition, they produce higher yields of crops. Identify some other types of modifications that would be useful for crop plants. Some examples could be resistance to insects and other pests, resistance to herbicides, resistance to viral infections, and foods that are resistant to rot and spoilage, or plants that produce plastics for the manufacturing industry. Many of these are already in production or currently in development. U.S. farmers have adopted GM crops widely since their introduction in 1996. Soybeans, cotton, and corn have been modified to tolerate herbicides and resist insect damage. The graph summarizes the extent to which these crops were identified between 1996 and 2012. Let's list some examples of GM animals that may be useful. Pigs can be genetically modified to produce more lean meat or high levels of healthy omega-3 acids. About 30% of the milk in U.S. markets comes from cows that have been injected with hormones made of recombinant DNA techniques to increase milk production. Using growth hormone genes, scientists have developed transgenic salmon that grow much more quickly than wild salmon. This effort makes it practical to grow these nutritious fish in captive aquaculture facilities that do not threaten wild populations. When scientists in Canada combined spider genes into the cells of lactating goats, the goats began to manufacture silk along with their milk. By extracting polymer strands from the milk and weaving them into thread, we can create a light, tough, and flexible material that can be used in such applications as military uniforms, medical sutures, and even tennis racket strings. Scientists are now using human genes to develop antibacterial goat milk. Researchers hope that cloning will enable them to make copies of transgenic animals, which would increase the food supply and could even help save endangered species. In 2008, the U.S. government approved the sale of meat and milk from cloned animals. Many farmers and ranchers hope that cloning technology will allow them to duplicate the best qualities of prize animals without the time and complications of traditional breeding. Biotechnology, in its broadest sense, has always been a part of medicine. Early physicians extracted substances from plants and animals to cure their patients. 20th century medicine saw the use of vaccination to save countless lives. Today, recombinant DNA technology is the source of some of the most important and exciting advances in the prevention and treatment of disease. Golden rice, which contains increased amounts of provitamin A, beta-carotene, a nutrient that is essential for human health. Provitamin A deficiencies produce serious medical problems, including infant blindness. There is hope that golden rice rich in vitamin A will help prevent these problems. Other scientists are developing transgenic plants and animals that produce human antibodies to fight disease. Several laboratories have engineered transgenic sheep and pigs that produce human proteins in their milk, making it easy to collect and refine the proteins. Many of these proteins can be used in disease prevention. Transgenic animals are also used as model test subjects in medical research, simulating human genetic disorders. These animal models have been used to study the onset and progression of disease and to conduct drug testing. Although they cannot completely simulate the responses of the human body. Animal models are useful and have been used to develop models for disorders such as Alzheimer's disease and arthritis. Human growth hormone, which is used to treat patients suffering from pituitary dwarfism, was once scarce. Human growth hormone is now widely available because it was mass-produced by recombinant bacteria. Other products now made in genetically engineered bacteria include insulin to treat diabetes, blood clotting factors for hemophiliacs, and potential cancer-fighting molecules such as interleukin-2 and interferon. This example will discuss how gene therapy can be used to treat someone who has defective hemoglobin. What would be the symptoms of defective hemoglobin? Well, hemoglobin is the protein used by red blood cells to carry oxygen. These individuals would not be able to effectively transport oxygen. So how could this disease be treated? You could give the patient hemoglobin injections or give the patient a healthy version of the hemoglobin gene. Gene therapy is the process of changing a gene to treat a medical disease or disorder. First, using biotechnology, we obtain copies of the normal hemoglobin gene. So how can scientists identify the gene for hemoglobin if they have the protein sequence? They can use the genetic code to identify the mRNA sequence, then use southern blotting to identify the gene. How can a scientist make many copies of this gene? PCR. Once they have successfully copied the gene, they place the gene into a genetically engineered virus. What else needs to be done before this virus can be given to a patient? The virus needs to be modified so that it doesn't cause harm. Next, they'll infect the patient's cell with the virus. In theory, the virus will insert the healthy gene into the target cell and correct the defect. The injected transformed patient cells are injected into the patient's bone marrow. The challenge is to deliver a gene that works correctly over the long term. For all the promise it holds, in most cases, gene therapy remains a high-risk experimental procedure. For gene therapy to become an acceptable treatment, we need more reliable ways to insert working genes and to ensure that the DNA used in the therapy does no harm. If two prospective parents suspect that they are carrying the alleles for a genetic disorder, such as cystic fibrosis, they can use genetic testing to find out for sure. The CF allele has slightly different DNA sequences from its normal counterpart. So, how can genetic tests use labeled DNA probes to distinguish the different versions of the gene? Well, like many genetic tests, the CF test uses specific DNA sequences that detect the complementary base sequences found in the disease-causing alleles. Other genetic tests search for changes in cutting sites of restriction enzymes. Some use PCR to detect differences between the lengths of normal and abnormal alleles. Genetic tests are now available for diagnosing hundreds of disorders. Scientists use DNA microarray technology to study hundreds or even thousands of genes at once to understand their activity levels. mRNA samples are isolated from two different types of cells or tissues, such as cancer cells and normal cells. Why might scientists study mRNA rather than DNA? mRNA gives an indication of which genes are active rather than simply which genes are present. In this example, cancer cells and normal cells have the same genes. The difference is which genes are actively producing proteins. Enzymes are used to prepare complementary DNA molecules from both groups of mRNA. Contrasting fluorescent labels are attached to both groups of DNA, red to one, green to the other. So what is the advantage to working with complementary DNA rather than RNA? cDNA is a more stable molecule. DNA fragments corresponding to different genes are bound to the walls in a microarray plate. Single strands of DNA are attached to wells in the plate. Each well contains a different gene, and all of the genes from an organism are represented in the microarray. The cDNA molecules bind to complementary sequences on the plate. So why are single strands, not double strands of DNA, attached to wells in the microarray plate? Single strands are used so they will bind to the single-stranded DNA labeled with the fluorescent colors. If the cancer cell produces more of a particular form of an mRNA, then more red-labeled molecules will bind at the spot for that gene, turning it red. If the normal cell produces more mRNA for another gene, that spot will be green. Where there is no difference between the two cell types, the spot will be yellow because it contains both colors. The labeled cDNA fragments compete for binding sites in the microarray wells. So if one of the labeled samples expresses the given gene more than the other, it will have more cDNA fragments than the other sample. Therefore, one of the samples will outcompete the other, bind to the complementary DNA in the well, and turn the spot either green or red. So, which spot in this image will turn yellow? And what does it mean? The spot in front will turn yellow. It means that both labeled samples expressed the gene equally. Which spot in this image links to a gene that is expressed more highly in cancer cells? The spot on the left is red, indicating a gene that is more highly expressed in cancer cells. How might a cancer researcher focus on research efforts on this gene? The gene is underexpressed in cancer cells. Perhaps it protects cells from cancer. The researcher could look at the product of this gene to find a protective protein. The complexity of the human genome ensures that no individual is exactly like any other genetically, except for identical twins. Molecular biology has used this fact to develop DNA fingerprinting for use in identifying individuals. DNA fingerprinting analyzes sections of DNA that may have little or no function, but that vary widely from one individual to another. Chromosomes contain many regions with repeated DNA sequences that do not code for proteins. These vary from person to person. Here, one sample has 12 repeats between genes A and B, while the second sample has 9 repeats between the same genes. Restriction enzymes are used to cut the DNA into fragments containing genes and repeats. Note that the repeat fragments from these two samples are of different lengths. What technique would you use to separate DNA based on this size? Gel electrophoresis. DNA fingerprinting has been used in the United States since the late 1980s. Its precision and reliability have revolutionized forensics, the scientific study of crime scene evidence. DNA fingerprinting has helped solve crimes, convict criminals, and even overturn wrongful convictions. To date, DNA evidence has saved more than 110 wrongly convicted prisoners from death sentences. This gel has DNA fingerprints between two suspects, S1 and S2, and a DNA sample from the evidence collected from a crime scene. So which suspect was at the crime scene? Suspect 2. In what sorts of crimes would DNA fingerprinting help in identifying the criminal or exonerating someone who is innocent? Cases where blood, other body fluids, or body tissues were left behind, such as rape or murder. What processes would investigators use to identify the herd from which illegally sold tusks of protected African elephants came? They would make a DNA fingerprint of each tusk for sale and then compare those fingerprints to those of elephants from specific protected African herds. In cases of disputed paternity, DNA fingerprinting makes it easy to find alleles carried by the child that do not match those of the mother. Any such alleles must come from the child's biological father, and they will show up in the father's DNA fingerprint. The probability that those alleles will show up in a randomly picked male is less than 1 in 100,000. This means the likelihood that a given male is the child's father must be higher than 99.99% to confirm his paternity. When genes are passed from parent to child, genetic recombination scrambles the molecular markers used for DNA fingerprinting, so ancestry can be difficult to trace. There are 2 ways to solve this problem. The Y chromosome never undergoes crossing over, and only males carry it. Therefore, Y chromosomes pass directly from father to son with few changes. The same is true of the small DNA molecules found in mitochondria. These are passed with very few changes from mother to child in the cytoplasm of the egg cell. Y chromosome analysis has been used in the same way and has helped researchers settle long-standing historical questions. One such question, did President Thomas Jefferson father the child of a slave, may have been answered in 1998. DNA testing showed that the descendants of the son of Sally Hemings, a slave on Jefferson's Virginia estate, carried his Y chromosome. This result suggests Jefferson was the child's father, although the Thomas Jefferson Foundation continues to challenge that conclusion.